Hi, I'm Scott Calvin, a professor of physics at Sarah Lawrence College, and you're looking at the pop-up book that I helped create. You can see the other two names down on there. Linnea Russell is a student of science and art, getting ready to graduate from Sarah Lawrence, and Bruce Rabel is involved with the Users Executive Committee at Brookhaven National Laboratory, which brings us to the subject of the book, the National Synchrotron Light Source II, Long Island's state-of-the-art X-ray microscope. This is a major new facility being built at Brookhaven. At the end of this video, I'm going to let you know how you're going to be able to make a copy of this book for yourself. But first, let's look inside. So, sometimes it needs a little straightening out. There we go. You can see that the first page is dominated by the shape of the building itself. This facility is actually a building size facility. The background image you see here is a Google Earth image of the facility. And there is a floor plan, I'll zoom in a little bit, of the facility. You can see these little spoke lines that come out. I'll explain what those are in more detail in a minute. But that's actually a floor plan of this facility. There's a box up here that tells us a little bit about it. Now, the facility generates X-rays, and we'll see what gets done with the X-rays. But if you take a look on the infield of a facility right here, it tells you how electrons are generated and accelerated and then injected into the ring. And you can see the information ends up continuing up along the interior of the ring here. So that gives an overview, but there's also some elements on the page that tell us in more detail about how X-rays are generated. For example, in this area we learned about bend magnets. Bend magnets are used for two purposes. One is they help make the electrons actually follow the shape of the ring. But the other is that when electrons are turned like that, they give off X-rays. So they become an X-ray source, which scientists will then use. Excuse me. So if I take my bunch of electrons here and send them around a ring, you can see that the result is the X-rays come out at the location of the bend magnet. That's one way to generate X-rays. Here's another. This device here is a representation of an undulator. It involves not just one set of magnets, but a whole array of magnets, spaced in such a way so that the electrons will oscillate, and as they oscillate, give off X-rays that reinforce each other. So the electrons here are shown in green, and they will be shown to oscillate as I pull the pull tab, giving off X-rays in purple that sequentially get more and more intense. So that's an undulator. Up here is a little booklet that's going to tell us about shaping magnets. So I'll open it up. Because electrons repel each other, as they travel around a ring, they would normally spread out. To keep them in line, there are quadrupole magnets shown here. But quadrupole magnets alone would allow electrons to spread if the electrons were slightly different energies. So sextupole magnets are also used to correct for the residual errors that the quadrupole magnets would leave. Turning to the next spread. So, give you a long shot here of the spread. So on this page, you see a representation of one of the particular beam lines, any beam line, in the NSLS-II. Remember each of those little lines I showed you on the previous page? If not, I'll flip back. These little lines in here. Each of these lines represents a beam line as depicted on this page. And you can see it's about 40 meters from the place where the beam line comes from behind the shield wall, the ring generating the X-rays is out here, up to where the experiments are done. 40 meters is about the length of 2 train cars. So you can see that's just one beam line. That gives you an idea of the size of the facility. So along the beam line here, one thing we can do is we can put together this first optical enclosure, which contains some optics to shape and focus the X-ray beam that's coming from the source. So first, there's a collimating mirror. The beam coming from the source is spreading out, and the collimating mirror brings it back so it's going more in a straight line. Then it goes through a monochromator. I'll talk more about that in a moment. And then when it comes out of a monochromator, there's a focusing mirror that brings the ray to a focus down where the experiment takes place. So I said I'd mention more about the monochromator. Well, here's the monochromator. So if I open up the monochromator, you can see there's some information about monochromators up here. And you can see this pair of crystals. So right now we are seeing the arrangement of crystals when a beam has a relatively low energy, when we want a beam to come out of a low energy. What's coming in here is what's sometimes called a white beam, a mixture of X-rays of all energy. But one particular energy will be right for this arrangement of crystals in this geometry. So what comes out will be one particular sort of color of X-rays, analogous to what happens when light hits a prism, and if you arrange the angle of the prism right, you could get red light to the spot on the wall. Or, if I change the arrangement, blue light. I could change to a high energy alignment. So that shows a little bit. You can see we get a different color out. I've depicted as a different color. Really, it's a different wavelength of X-rays, but it's analogous to color. Okay, so that's our monochromator. Okay. Tricky to do these things one-handed since I'm holding the camera. There we go. All right. Now we come over to the really exciting part. We get to do an experiment. Not just one experiment, but we can pick from 3 different kinds of experiments. All of these kinds of experiments are done at synchrotrons such as the NSLS-II. So let's take them one at a time. So first, let's take ZAF spectroscopy. So there's some information here about ZAF spectroscopy. And it tells you some of the equipment that you need. So where do we get that equipment? Well, in our case, we get it out of this pouch. So let's see. Again, one of these things a little tricky to do while holding the camera. There we go. So one of the things that's in the pouch is a picture of what an actual ZAFS experiment looks like with the components labeled. And here I pulled out our papercraft version of the components. Now sometimes they get a little mashed while they are in the pouch, but this is a special kind of, it's not actually paper, it's a special kind of plastic. And just by pushing at it, I can kind of unmash it a little bit. And once again, trying to do it one-handed. I may set the camera down a minute. Let's see if I have to. No, I think I did okay. All right, so where are experiments done? They're done in this end station over here. You can kind of get a sense of a scale from this picture of Bruce on the back. That's about how big these end stations are. So this is a small room. And in the small room, you can see I've got part A. So I can take part A, and I can slide it in where the A is, following the arrow. try to get that. That's pretty much shaped right. And now I've got a part B, somewhere on there, B. And I can slide that in where the B is. And I've got part C. By the way, they're each labeled. This is an incident detector. And I can slide that in where the C is. And now I complete the shape of my end station by sticking the orange tab in to the orange slot. It's gotten a little distorted while the book was closed. I can bend it back into shape, maybe a little bit like that. Now the distortion's gone. But I knocked over my transmission detector. There we go. Come on. Tis tricky to do this with one hand. Almost there, almost there. Ta-da! Okay. So, there's my setup. It probably looks better from the side in this case. Okay, so we've got our setup for the experiment. So once we have the experiment set up, of course, we'd like to conduct it. And we'd like to look at our results, which are in the logbook. So I open the logbook, and the first page talks about ZAF spectroscopy, the kind of experiment that we're doing. And it gives you an example of such an experiment. And here it shows you what the data might look like on the computers as we collected it using a setup such as the one we just made. And when I lift this, we can see an atomic structure, which is the structure that that data would tell us is present. So by setting up that kind of experiment, we could collect that kind of data. By analyzing that kind of data, we could get information that the atoms are arranged in this way and therefore understand whatever the material was better. In this case, a material that acted as a solar cell. So that was nice. That was ZAF spectroscopy. Let's see what else we can make. So I'll set the ZAF spectroscopy aside. Let's go to macromolecular crystallography. You'll notice they're all color-coded. This is color-coded red. Let you read that if you want. So let's see what's in here. Good stuff in here. Not that ZAF isn't good, but these are a little flashier looking. And should be an insert, yes. So there is what an actual molecular crystallography, MX experiment looks like, with various components labeled. And here are the components. So which one's A? This one is A, lowercase a this time, in case you're colorblind. It still lets you figure out which colors are which. And coded red. Get the shape back. I have the shape back. And over here you can see there's a place for A. So I will put in A. There we go, got it. Okay, so we have the detector in. What else have we got here? We have B. B goes in an exciting place. B goes, see those slots labeled B? Let's put them in there. Ta-da, one goniometer on the wall. And, C. There's a slot labeled C. That cryostream is a little floppy. I can put it in different places. So the question might be, where do I want to put it? It actually has instructions on the back of it that says point cryostream in direction of goniometer tip. So I can do that. There it is, pointed at the goniometer tip. I should complete my end station in the usual way. And let's see if I can get an angle here where you can see everything. There we go. So there's my experiment. So now I have a molecular crystallography experiment. So of course I can see that data as well. Learn about it a little bit. There's information on this page about it. And on the right side is a diffraction pattern, the kind of thing that comes out of a molecular crystallography experiment. So it says lift to see structure. Ooh, this is going to be particularly challenging one-handed. Let's see what I can get here. Okay, got it in focus. I lift. Ah, nice. Okay, and you could see the structure of the protein channel underneath. Okay. One more kind of experiment. We'll set these aside. Done with our molecular crystallography experiment. Sorry for a little jump there. As I said, done with the molecular crystallography experiment, and the one more is the imaging experiment. So, I'll let you look a little bit at the imaging experiment. You can pause the video if you want. And let's see what we have in here. Oh, this is particularly exciting stuff. Look at that. Oh, geez, I think there it is. Okay. So, as usual, picture of the actual experiment. And labeling what the pieces are. Let's see what we have over here for the components. Which one's first? That is a 2. That one is a 3. Aha, that one's a 1. Alright, so let's put this plant here, which we're going to study, into the 1 slot. There we go. And now let's get piece number 2, which was the KB mirrors. So I have here the KB mirrors, and they go into slot number 2. There we go, that's the KB mirrors, and now the silicon drift detector. That will go into slot number 3, following the arrow. There's slot number 3. And finally, the optical camera. As with the cryostream and EMX experiment, I've got to put it in the right slot and then point it towards, put it in the right slot, and then point it towards the sample, which in this case is the plant. That's pretty much got it. So, there is my experimental setup. I suppose I'll assemble the end station, and there we have it. So, let's see what the logbook has to say on this one. So I open up, past the MX experiment. Oh, here's the imaging experiment. So this tells me about the imaging experiment. And over here I can see the results. I have this photograph, essentially, of the leaf, which is being imaged. And then I can look at the cobalt concentration in the leaf and the calcium concentration in the leaf. Very different distributions. And of course, both together. Okay. So that's our experiments. I will clean up after ourselves. By the way, I should say that in the actual beamlines at the NSLS-II, they're usually optimized for one of these types of experiments. In fact, each beamline is specialized for particular purposes. They can still do a wide variety of materials, a wide variety of detailed experiments. But, for example, the first optical enclosure may have some different details in order to optimize it for the type of experiment. The way the end station is laid out will generally have these kinds of pieces of equipment semi-permanently installed. They're heavy, complex pieces of equipment compared to the papercraft we have here. Some of them less so than others. This particular piece, for example, is fairly easy to move around. But some of these things, like the goniometer, are likely to be essentially permanently installed. So the swapping out here one to the other is really representing what would happen if you went from one beamline to another at the NSLS-II in general. Although there is some ability to change some of the setups involved. Okay, so let me close all this and finally move to the back cover. Okay, so I've moved over to the back cover. So the back cover tells you a little bit more about the book. It tells you one of the systems of the text in the book. That the blue text tells you that you can find more information on Wikipedia, for example. A little bit about the components of the book. They're actually a sort of plasticky material, mostly, not paper. Some of the information on some of the units that are used to give you a feel of how big they are. Set of acknowledgments of all of the wonderful people who helped give permission for materials in this book. The people who were the primary contributors to the book, Bruce, Linnea, and myself. And importantly, the license for the book. Except for 2 photographs, which are listed here, the license for the book is a Creative Commons Attribution Share Alike license, which means that you can take the book and modify it in any way you choose. You could even use it for commercial purposes, as long as you continue to give the original creator's attribution. The details can be found in the license online. This video is also under the same license, so you can use clips of it, whatever you'd like to do, as long as you give us proper attribution under this license that you can find here. Now, the exciting part is that we are going to have online at bruceravel.github.io/synchrotron_/pop/ I'm sorry, you can see it. bruceravel.github.io/synchrotron_pop_up_book. We are going to have all of the image files needed to make this book, along with instructions how to do so. You'll have to get your own materials, but you will be able to make a copy of this book for yourself. Once we get all that up there, and it's not up there yet as of the time I'm recording this video, I expect by around July of 2014 we should have all of that up. You would have to get your own materials. You could do it with paper and ordinary materials like double-sided tape, but we'll also have links to where you can get the special plasticky kind of paper and that sort of thing. The assembly, doing it on your own, is the kind of thing that would be good for a rainy weekend afternoon. Won't be able to do it in 5 minutes, but you'll be able to do it in a few hours. And then you can have a copy for yourself. Well, thank you for taking a look at this book with me.